Thanks. Our next award is presented to a basic science paper entitled A Multiscale Structural Investigation of the Annulus-Endplate Anchorage Systems and Its Mechanisms of Failure. This paper will be introduced by Dr. Neil Broom and then presented by its lead author, Samantha Rodrigues. Dr. Broom?
Well, look, firstly, I'd like to express on behalf of all three authors our gratitude, our sincere thanks to the Awards Committee of NAS for recognising our disc research back in Auckland, New Zealand. It's a long way from here. So firstly, here are our disclosures. Now, as a brief introduction to our presentation, one of our primary goals has been to develop novel approaches aimed at understanding better the structure-function relationships governing disk behaviour. And in this first slide is an example showing transverse stretching and simultaneous imaging of what is ostensibly a simple crimped array of annular fibres. And it reveals a level of structural complexity that would probably be missed if we use conventional histological approaches. And the key here in this kind of experiment is to maintain the tissue in its fully hydrated state while actually doing the stretching. Secondly, this is another example where we've taken a rather unusual approach of taking out or extracting a core of disc vertebra from an intact motion segment and then ring barking any remaining annular elements. If we do that, the nucleus can be shown to stretch 4 to 5 times the original disc height before finally rupturing under a significant load. And this kind of experiment provided clear evidence of a definite integration across the nucleus end plate junction. And this is achieved in part via a mechanism that gathers up the nuclear fibres into a series of nodes that insert into the collagenous end plate. And one such node is shown here in this slide, imaged optically and under SEM. This structural integration also relates nicely to our work on disc herniation, which we're currently doing. And this slide shows how under combined flexion and compression, the extruded nuclear material has taken with it some end plate with it, an observation that is consistent with some clinical reports noting that herniated nucleus that is extruded may at times include attached end plate tissue. So at this point, it leads nicely into Samantha. Now, Samantha is one of our PhD students working in our research group, and she is in her final stages of her PhD, and she's going to present her work on the micromechanics of end plate failure. So, Samantha.
Thanks, Neil. So disc herniation frequently involves failure of the end plate junction, but little is known about this critical biomechanical anchorage site, especially at the level of its complex microarchitecture. The aim of this structural study was to better understand the failure mechanisms of the Annulus-Endplate junction, the hypothesis being that certain modes of loading would make it more susceptible to failure and thus intimately associated with disc herniation. So for our failure experiments, ovine motion segments were loaded to failure in torsion, oblique tension, and axial tension as illustrated. We focused on the anterior annulus for ease of access to the junction. Finally, structural failure was assessed using DIC optical microscopy and scanning electron microscopy. Now, if you choose a sectioning plane carefully, this is what the junction looks like intact in its fully hydrated state. The anchorage itself is rather shallow, so the annular fibers penetrate through the entire depth of cartilaginous end plate, most of which is calcified, down to the bone junction where they appear to end. Within that calcified zone, however, the main bundle branches into several sub-bundles, a mechanism that we believe strengthens this anchorage site by increasing the area over which forces are distributed. In terms of failure of the system, we observed 2 main modes. The first was failure at the tide mark, which is the boundary between the soft annulus and calcified end plate. and the second was failure at the cement line, which is the boundary between the calcified end plate and bone further down the anchorage. And here's the frequency of the anchorage failures for the 3 different loading types. The numbers represent the number of samples in the table. So compared to torsion and oblique tension, the samples tested in axial tension contain a greater proportion of anchorage failures, particularly at the tide mark. Although we tested the anterior annulus, the same principles would apply to the posterior region when stretched in flexion, and thus these findings are consistent with previous studies showing that flexion increases the risk of herniation involving end plate failure. The question is why? Well, we need to consider how these annular fibers respond to the applied loads. In torsion, when the disc is rotated, only one set of fibers, those aligned in the direction of twist, is stretched, and more or less along their natural oblique angle. The same happens in oblique tension. In axial tension, however, both sets of fibers are pulled vertically. This causes them to bend sharply at their insertion into the calcified end plate, a soft hard tissue junction, creating high stress concentrations here and eventually leading to tide mark failure. But what happens if we artificially remove calcification? To find out, we repeated axial tension tests with samples decalcified for varying periods of time prior to loading. Now, we know that this is not a natural state, but the purpose is to understand the role of calcification here. And the effect is dramatic. So our fresh samples were typically dominated by mid-substance annulus failure. A short overnight decalcification, however, leads to considerable failure at the darker anchorage surface. And further, microscopic examination of these decalcified samples confirmed that failure occurred at the cement line and across several bundles rather than the individual bundles seen sometimes in fresh samples. So the following schematic helps to explain what's going on. Our decalcification pre-treatment process essentially removes the strengthening minerals from the end plate and bone too to some extent. This softens the end plate so that acute bending now occurs further down the anchorage. The resulting scale of cement line failure demonstrates how important calcification is in strengthening this junction, particularly across the cement line. An interesting observation, though, was that in many of our samples, even those decalcified for 2 weeks prior to loading, small segments of bone were extracted still attached to the annular subbundles and cartilaginous end plate. Now, if calcification alone was responsible for keeping the cement line intact, then surely a 2-week decalcification would have produced a clean separation at this interface. Perhaps there's a finer level of integration involved. Using a higher resolution microscope, we examined the intact anchorage site in its full fibrillar detail. So here's an image taken from the anchorage site. And in this image, you can clearly see the finer fibrils that form the alternating leaves of annular subbundles and cartilaginous end plate within that anchorage zone. We can also follow these annular subbundle fibrils further down to the cement line and the vertebral bone to see how these two tissues are connected together structurally. So taking a closer look, we found instances where the annular fibrils penetrate a short distance beyond the cement line, and areas where the annular fibrils merge and integrate with the bone fibrils, as highlighted in the boxed regions. The annular fibrils also merge with the cartilaginous end plate fibrils in a similar way. Although short-ranged, we believe that these structural connections help to strengthen this junction and more likely account for its remarkable strength and tension compared to just calcification alone. So to conclude, we found that the Annulus-Endplate junction is strongly dependent on both calcification and fibril level integration. It is more vulnerable in axial tension due to acute bending at the soft hard tissue junction. And this possibly explains why flexion increases the risk of herniation involving end plate failure. Finally, we would like to thank the North American Spine Society and the Spine Journal for the Outstanding Paper Award and this opportunity to present our work here. I would also like to acknowledge the University of Auckland for the doctoral scholarship which has funded my PhD. Thank you.